2, 1. Good morning, my name is Dominick.
And my name is Yarali Hidalgo.
Today we are presenting the effect of salt concentration on potato osmosis and weight change.
So for an introduction of our presentation, the experiment is going to investigate how different concentrations of salt affect the process of osmosis in potato cells. By measuring the weight change of potato pieces after being soaked in the different salt solutions, the aim of the project we are doing is to understand the relationship between salt concentration and water movement in and out of potato cells. The findings will provide insights to the principles of osmosis and its practical implications in biological systems in real life. The study will also be able to highlight the importance of controlled variables to ensure accurate and reliable results. So for an overview of our presentation, we're going to hit different topics, starting with our background research. We developed a lot of background research in order to actually help us go through with our project so we know what has to be known. Then we move on to our hypothesis and question. This is going to include our general hypothesis and our general question of our overall experiment. Then we go into methods. Methods will be a step-by-step instruction of our experiment and our general objective. After that, we're going to hit results. Results are where we're going to discuss the results and actually show what happened throughout the time in the project. Then number 5 is discussion and conclusion. We're going to summarize the findings of our experiment, and then we're going to see if they lined up with what our hypothesis was at the beginning. And then we're going to hit our references, which were what we found our research from.
Our background research. Number 1, what is osmosis? Osmosis is the movement of water molecules through a semi-permeable membrane from an area lower solute concentration to an area higher solute concentration. Cell structure. The cell membrane controls what enters and exits the cell, while the cell wall provides a support and structure. Cell structure. Plant cells have a cell membrane and a rigid cell wall. How does osmosis play? Osmosis is the movement of water through the cell membrane, helping maintain turgidity in plant cells.
Number 5, what are potatoes made of? Potatoes are made of cells that contain water, starch, and various solutes. Whenever you place potato slices in different solutions, like salt water or the pure water we're using, osmosis can occur. The structure of potato cells. Potatoes are primarily composed of many different cells which are specialized for storage. These cells contain large vacuoles filled with water and starch. The starch serves as an energy reserve for the potato itself. The temperature effect on osmosis. Water temperature can increase the rate of osmosis as molecules move faster, which could lead to more significant changes in the potato slices. That is why we chose to keep our potatoes at 73 degrees Celsius. Fahrenheit. Tonicity describes the ability of a solution to change the shape or tone of cells by altering their internal water volume.
Our background research. Isotonic solutions. An isotonic solution has the same solute concentration as the cell. Hypotonic solutions. A hypotonic solution has a lower concentration as the cell. Hypertonic solutions. A hypertonic solution has a higher concentration as the cell. A semi-permeable membranes. Semi-permeable membranes are barriers that allow certain molecules, like water, to pass through while blocking larger ones. They have tiny pores that determine which substances can cross, enabling osmosis, where water moves from areas of lower solute concentration to a higher concentration to balance the levels. Question 9, hypothesis. Question 1. Our question is, how does saltwater affect potato cells? Question 2, or hypothesis, if potato pieces are submerged in salt solutions of different concentrations than the pieces in the highest salt concentration, 10%, lose more weight and feel softer compared to those in lower salt concentrations, 0% and 5% because of osmosis. Materials are potato, the main factor of the experiment, water to use to mix with the salt to perform the experiment, the scale to measure the weight before and after the experiment occurs, salt to mix different values with the water and to submerge potato, cups to hold our solution and submerge potato, knife to cut the potato into 3 chunks.
So the method and variables to be considered are the independent variables, which is the type of solution those slices are placed in. So we're using plain water with no salt, 5% water, and 10%. The dependent variables is the change in mass or texture of the potato slices after they have been soaked in the different solutions. And then the control variables that will stay the same throughout the whole time of the project is the size of the potato slices, the temperature of the environment, the time the potato slices are left in the solution, the volume of water used, and the shape of the potato. Our methods, objective and procedure. The objective is to observe and record how the salt water can go and affect the potato cells. The procedure, we took 3 cups of water that were 75 degrees Fahrenheit and left one with no salt with 1 cup of water. We left another one mixed with 5 grams of salt and put it into another cup of water, which is our 5% solution. We mixed 10 grams of salt with 1 cup of water, and that made us our 10 grams solution. We cut the potato into 3 1-inch by 1-inch pieces and weighed the pieces before and documented the weights. All of the potatoes were the exact same weight at 12 grams each. The procedure is we're gonna soak the pieces into different solutions and let them soak for about 30 minutes. We are gonna remove the potato and feel the potato for any differences in texture. Following that, we're gonna weigh the potato again after taking them out, record our observations, and discuss how the salt water went on to affect the potatoes.
The results. So here's the before results as we had our potatoes out and all the water that had the salt on it. This one has 0, this one has 5 grams, and this has 10 grams. This is during where we submerged the potatoes and put a timer on for 30 minutes. This is after whenever we stop the timer after 30 minutes, and this is the potatoes out of the cups.
So for the results, all of our potatoes started really firm and not easy to squish through, and they all started weighing at 12 grams each. So potato 1 was submerged into 0% salt, potato 2 was into 5, and potato 3 was into 10. After the 30 minutes occurred, we took them out and drained them and let them sit on some paper towels for a little bit, and we decided to fill the potatoes. The feeling of potato 1 stayed the exact same. It was really firm, we couldn't squish it. After sitting in the salt for potato 2, it was somewhat firm and it was a little bit squishy. It wasn't as squishy though as potato 3, which was soaked in the 10% salt. For the weight of our potatoes, potato 1 stayed the same as it was in the beginning at 12 grams. Potato 2, on the other hand, it lost 1 gram in weight after it was stayed in there for 30 minutes. Potato 3 lost 3 grams of weight and it weighed in at 9 grams at the end of our experiment.
Our discussion and conclusion. In our experiment observing osmosis in the potato cells, we have discovered that the potato being submerged with the most grams of salt, 10%, lost the most weight and felt the most different compared to the initial starting weight and feeling. The cups with no salt or less salt had less or no change in results.
So for our broader impacts, this experiment helped a bunch of different impacts, and it had a bunch of different ways that it could impact society. So the osmosis experiment with potato cells had interesting impacts that touch on everyday life. It starts off with medical applications. Understanding osmosis is very important in medicine, especially when it comes to things like IV therapy. When doctors give patients fluids, they need to make sure the concentration is just right so that the cells don't shrink or swell, which could cause serious health problems later on in the future. Agriculture. Farmers can use the principles of osmosis to manage how they water their crops and deal with soil salinity. Knowing how salt affects plant cells helps them grow crops more effectively, which can lead to better food production and security. Food preservation. Osmosis is a key part to preserving food, like when curing meat with salt or making pickles. By controlling the water content in food, we can keep it from spoiling too quickly, which means less food waste and longer lasting groceries. Environmental science. Environmental science. Osmosis helps us understand how plants and animals adapt to their environments, especially in places where the salt levels in soil or water are changing due to climate change or human activities. This knowledge is important for conservation effects and creating sustainable practices. By this experiment, we're not just learning about osmosis, we're also getting insights that can help in fields like healthcare, farming, food tech, and environmental science.
Our strengths and weaknesses. Strengths. We had the opportunity of having the proper tools to conduct this experiment. Example, the thermometer, the scale. During doing appropriate research before conducting this experiment. When we did this experiment, we were having trouble to find the information, but we found some, and it helped a lot during the experiment because it made it more easier. The weaknesses: we could have used more substances other than salt to see results from others. Example, sugar. We could put sugar in 5 grams in the water and 10 grams into the water to see how it is. We could have had done more trials with different percentages of salt added. Example, 15% and 20%.
These are our resources that we used for our project, and that is the end.